just introduce our two presenters. I'm super excited about this opportunity. It's in honor of World Ocean Day, which is today, June 8th, and also the One Mississippi River Network. They have their River Action Days, and it's a huge network with the Mississippi River watershed. It's a bunch of different organizations and groups that work together to communicate and share activities that are going on in the watershed. And so today we're here at Marvet, Mississippi with a group of veterinarians and veterinary students and others that are in the field. And this particular presentation is on the One Health concept. And we have Dr. Ruth Carmichael, and we also have Dr. Jenny Bloodgood from Cornell. And so I'll let them just briefly introduce themselves, and we're super excited. Thank you for joining us today as well.
Great. Yeah, thank you so much for having us. So we are going to truly kind of tag team this. So I actually kicked off my shoes because if I didn't and we're moving around, I'm going to trip. So forgive me, but I just got 2 inches shorter. So I am Ruth Carmichael. I'm a senior marine scientist at the Dauphin Island Sea Lab, which if you're not familiar, Dauphin Island is one of the barrier islands that separates Mississippi Sound from the rest of the Gulf of Mexico. And yeah, sorry. So we're on one end of that island, and so that's where we're located. I'm also a professor of marine and environmental sciences at the University of South Alabama. So I have been in the region for about 20 years now, and most of that time have been working with manatees and also now other cetaceans in the area. So yeah, we're going to start off. Well, I'll let you introduce yourself, then not wait till your section.
No problem. So I've already introduced myself to the room, but for everyone on Zoom, I am Dr. Jenny Bloodgood, and I am a veterinarian based now at Cornell University. So I work with our New York State Department of Environmental Conservation as their wildlife veterinarian. And then I used to be the veterinarian for the Alabama Marine Mammal Stranding Network, which is based at Dauphin Island Sea Lab, working with Ruth on stranding responses there. Nice to meet you all.
Great. And so I'm going to start off with something a little interactive here. Sorry, everybody on Zoom. I don't know that you'll be able to see what's going on, but I'll talk you through it. But I just would like to see a show of hands. And when you raise your hand, just leave your hand up. But how many of you all live on the water? Anybody here from the Gulf Coast? A couple? Or no, what's okay if you're not on the Gulf Coast? It's okay. A lake, a river, a stream. Yeah, okay. And so how many of you all, anybody fisher persons? I'm not going to leave your hand up. If you already put your hand up, leave it up. Fish. All right. Anybody scuba dive? All right, we got a few more. All right. Eat seafood. All right. Like to watch wildlife. Yeah. All right, I think I finally got everybody. All right, so we all can appreciate this coastal environment that you guys hopefully have had a chance to spend some time in since you've been here. We have about 600 miles of coast in Alabama. We have about 400 miles of coast in Mississippi, and together it represents about a $16 billion coastal industry for our region, which is pretty huge when you think about it, right? But unfortunately, despite, oh, that's the wrong computer, this awareness of the value of our coastal systems and how much, you already just told me how much you all value it. And I love my seafood, as you can tell, I've got a seafood belly, right? But there are a number of threats to our coastal systems, right? And some of these you are probably already quite familiar with. The first, of course, that we often think about is changes in land use and land cover that result in changes in hydrology, for example, and the rate at which water is delivered to coastal systems. So this can alter freshwater delivery to the systems. And I think you've talked a little bit about freshwater delivery earlier. We're gonna revisit that a little bit as well. We'll do a little overview for folks on Zoom who might not have seen that other presentation. Also, with this freshwater conveyance, we have changes in erosion and sedimentation. We have delivery of contaminants. We also have direct delivery of debris, right? So all these things, unfortunately, you've probably seen. Also changes in nutrient loads, right? That can deliver or change the quantity of noxious or harmful algae that are delivered to our waterways. And extraction processes. So fisheries is obviously a real common one. We have to worry about over extraction. And then finally, of course, simply the presence of people and human wildlife interactions, which we're having growing awareness is, a real concern. So one of the kind of classical, I teach marine conservation biology, and the classic idea that we've sort of built conservation science on over the last several decades is this idea that we know that things people do on land and in the water can affect our coastal systems, can affect our oceans. But in recent years, what's been really interesting is we've started to think a lot more about what an altered ocean means to our human health. Okay, and there is actually a growing body of literature where they've actually studied these questions. And we know that our mental health and physical health are linked to the well-being of our coastal waters, right? So we're getting growing awareness of that. But how does that really work? What are those real links and what kind of threats are we talking about then? We were talking about threats to the environment, but what are the links to the threats to public health? Okay, so we're going back to the same kind of idea. So with that increased conveyance that I talked to you about, so for example, those changes in freshwater, changes in urbanization and land use, we can have direct and indirect effects. And one of the direct effects is you can get increased conveyance of microbes, right? Pathogens like bacteria and viruses and really wonderful nasties like zoonotic diseases that you guys are pretty familiar with. Also, because of that, we can have links to food insecurity in this case, because when you have the delivery of these microbes to coastal systems, you often have closing of shell fishing areas. So we have limited access to seafood and also closing of swimming areas. So we have then restricted recreation, right? And I just showed you a slide that said we have increasing evidence this is all linked to our well-being, right? So already we're talking about direct health effects and also mental health and well-being. And then, of course, we have the wonderful contaminants, things like metals. And we're going to talk today a little bit about forever chemicals, so things like persistent organic pollutants that we have to be particularly concerned about because not only are they contaminants, but they're contaminants that literally persist in the environment. So we have to worry about longer term wildlife and human health effects. And then, we need to be thinking, of course, about health, but also, this doesn't just affect our physical and mental well-being, it also affects our economic well-being in our coastal areas where it's affecting our ecotourism. And I gave you that statistic at the very beginning that we have this multi-billion-dollar coastal economy. Well, if you look at the cost of health, the cost of treating persistent long-term health problems, it's actually in the trillions of dollars, right? So we took an effect, a multi-billion-dollar effect on the economy and just put it into the trillions by showing you a link to human health. All right, so all of this idea that I've just been talking to you speaks to these connections between environmental health, animal health, and human health. And that's the underpinnings of the One Health concept. This idea that what we do in the environment that affects animals, that affects food resources, necessarily affects us, right? This idea of these interconnections between people and organisms in their shared environment, right? And because this is already, you can see, hopefully so multidisciplinary, it necessarily takes collaborative, multisectoral, and transdisciplinary research approaches to come up with the similarly scoped types of solutions. So how are we getting at this? And so our model that we're going to talk to you about today, we're going to present some case studies using marine mammals. The great thing about marine mammals is, these animals are our closest human analogs in coastal waters, right? Many of you probably already know that. They literally sort of live where we live, right? If you're living on the coast, they swim where we swim. They eat the same foods that we eat, which means they're excellent sentinels of human health and coastal systems. So we're going to present some case studies today, but I wanted you to understand a little bit about where those kinds of data are coming from. So Dr. Bloodgood mentioned that we have the Alabama Marine Mammal Stranding Network that's in Alabama. Dr. Deb Moore spoke to you earlier, working with IMMS, which does the same kind of work here in Mississippi. But what we are doing is stranding response coupled with research to collect the kinds of data that let us answer those kinds of questions that get at these links, these multidisciplinary links to human health. So for example, we do necropsies, which you guys all know what necropsies are, right? This is the one group, I probably don't have to explain that to you. Histology, we might send samples out for disease analysis, some of that microbiology. We even do a lot of contaminants analysis in-house, but we also can send some out, biotoxin analysis. And then we also do a whole suite of ecology studies to help us understand how the animals are responding to whatever's going on in their environment, whether it's natural or it's human induced, right? So we're looking at all different kinds of ways of analyzing how these pieces are all fitting together. So I also wanted to show that it does take a village to do this. So it's not any one group working on these kinds of projects alone. And so when we go through the different case studies today, I really want you to think about the fact that regardless of who the stranding response organization is that would be collecting these data, pulling the pieces together, we have a number of funding agencies and also teams who literally work around the country that help us with all of this work. So with that, I will turn it over to Dr. Bloodgood to start us off on our case studies.
Okay, so we are going to now walk through just a couple of case examples of the types of things that Ruth mentioned. So the first, we have a manatee case study, and I can't do the pointer.
I forgot. All right, it's my bad. I teased you. I forgot. I wanted to mention that, I mentioned that whether it's natural effects or environment, human effects. Obviously, the foundation of this is we have to understand where those factors are coming from, where those effects are coming from. So there's actually, I just want to give a nod to a lot of the kinds of research that are being done, not just by folks who do marine mammal work, but in all of our facilities. And especially, this is just an example of the kinds of work we do at the Dauphin Island Sea Lab. But my lab and colleagues are working on understanding those freshwater injury, understanding what's happening with plastics and microplastics, and a variety of contaminants. We do microbial and pathogen work, and even a number of other indicators that are indicators of human influence in coastal systems. So I just kind of wanted to give a nod to this and also show Mobile Bay. Dauphin Island Sea Lab is down here. Because just this one embayment, thinking about the whole Gulf Coast or any of our coastal areas, you can kind of get a sense of just a tiny little bit of impact, right? And this is huge. All the sources of potential contaminants, wastewater, agriculture, human residences, birds and wildlife, and then all the resources that we have associated with this. And one thing I also did want to point out is Mobile Bay is the fourth, well, depending on the statistic you look at, the third or fourth largest freshwater drainage in the country among watersheds that are in the United States. And if you look at the amount of freshwater flow we have coming through our system relative to the size of the watershed, we actually rank #1 in the country. So we have a huge amount of freshwater discharge coming down. And then you think about the fact that just over here is Mississippi, right? And they've got the largest discharge in the country. So we have a tremendous conveyance of all of those things that I talked to you about in the first few slides. So I just wanted to set that stage. Now, I'm sorry, Jenny, go ahead.
That was beautiful. Okay, so we're gonna jump into a manatee case now as an example of a One Health approach to a multi-factor mortality in this animal. So this is not the animal I'm going to talk about today, but this is just an example of a manatee that our group responded to back in 2010. So the manatee that we're going to talk about today, so manatees typically range from the Florida Peninsula, but all the way out to Texas and then up into North Carolina, even up to Delaware Bay. So a lot of people think manatees, and we talked about this earlier, you automatically think, what state?
Florida.
Florida. Everybody thinks Florida. But really, they range much farther, especially in the warmer months. But they need to go back to Florida or find a warm water refuge during the cold weather, because what is their temperature that they can't go below for long periods of time? That was great, guys. Yay. We're learning things. Yeah, Celsius. It's actually easier to remember in Celsius. So around 20 degrees Celsius, and which is about 68 degrees Fahrenheit, is the thermal tolerance of manatees. So if you start to get below that range for long periods of time, especially, manatees can start to experience what's called cold stress. So that's one of the major threats to them outside of their Florida range. So if they come to Alabama in the warm weather, it's really nice, they love it. But if they stick around during the winter, they can experience cold stress if they don't go back to warmer waters or find, as I mentioned, a refuge that has warm water. The other threats that they can experience throughout their range are boat strikes, and we have a slide about that in a little bit. Habitat loss, just like all wildlife species, and then entanglement and ingestion of marine debris is a major issue. So this manatee that we're going to talk about today stranded while I was still working at Dauphin Island Sea Lab, and we do responses for manatees both in Mississippi and Alabama. And this manatee stranded in Theodore Bay, which is actually not far from where the Sea Lab is. And we got a phone call, on a January morning. So automatically you're thinking, it's cold. Yeah, it's cold. So we got a phone call about a manatee in Theodore Canal. So the first thing that we did was immediately deploy a team to go look at the manatee and assess the situation, assess what we can about the body condition of the animal, how it's behaving in the water, and do we see any potential cold stress response in this animal. So I'll tell you first off that the water temperatures, that's a great way to just immediately, you can jump online, even you don't have to go measure it directly, but there are stations where you can measure it. It ranged in the weeks preceding this call about this manatee from about 10 to 15 degrees Celsius. So we already know that it's been cold for quite a while for this manatee. If you look at this manatee based on our talk earlier about manatee condition, what are you thinking? Yes. That's a peanut head. Yes. So for everybody online, we're talking about this little depression behind the skull in the animal. Manatees should be nice and plump and round and be shaped like a cigar. And this manatee has lost some of the fat behind its head. It's called the nuchal fat, and that can lead to this depression, and it makes the animal look like it has a peanut for a head. So we call it a peanut head. But that's telling us immediately that this animal is stressed in some way. So it has lost some of its fat stores. And so just from that alone, we start thinking this animal is probably not doing great in winter here. We're also noticing maybe a little bit of bleaching along the snout in the extremities, which is another sign of cold stress. So based on this information, we started coordinating with, do you guys remember the federal agency that oversees manatees? This was a tough question. Fish and Wildlife. You guys are great. Okay, so we're starting to coordinate with Fish and Wildlife and then other organizations that do a lot of manatee responses. So SeaWorld and the Florida Fish and Wildlife Conservation Commission. So we are coordinating with these groups to start talking about a potential rescue for this animal. So the idea is that this animal is not going to make it back to a warmer climate on its own this winter. It's already in decreased body conditions, so it's undergoing some chronic stress already at this point. So we started working on a plan to rescue the animal and transport it to a rehab facility. Unfortunately, this is not a happy ending for this manatee. It died after capture, during transport. So we were able to get out on the water, assess the animal, and we did that for a couple of days before the rescue. But during the rescue, the animal, prior to us approaching it, we watched it from far away. That's your first step, see what's going on. It was tachypneic, so it was breathing really quickly and had some erratic behaviors, and it stranded itself on the beach. It had been in deeper water, but stranded itself on the beach twice, right before we went in to collect the animal and try to get it onto a transport. So this animal was already very, very stressed, and we were able to collect the animal, put it onto a boat, kind of like I described earlier, drive it back to where we had the transport van, which was a pretty short ride, but maybe 5 or 10 minutes. During that time, we monitored the animal, and it was doing okay. It only breathed a couple of times, but that's not abnormal for such a large animal. And we got the animal into the truck and started off we were going to Audubon, weren't we? Yeah. So we started off towards Louisiana. But unfortunately, after about an hour, we lost the heartbeat, we lost respiratory rate, and even with emergency medical intervention, we couldn't save the animal. So it died during the transport, but it was already in the truck. So the best thing that you can do to learn more information about why this happened is to do a necropsy. So cause of death determination, like Dr. Carmichael mentioned. So we basically turned around and went straight to Dauphin Island Sea Lab to do a necropsy of this animal. So based on the necropsy, first you do a full external examination. And I want to make sure everyone on Zoom knows that there are a couple of pictures coming up. They're not super graphic, but there are some organs on the slide. So I'm used to talking to vet students who are like, no big deal, that's fine. So just so you know, there's like semi-graphic content coming up. But okay, so based on the external exam, we did see, you can see in the yellow circles on the top right image, the skin bleaching was confirmed. So you can see how white the extremities are, the extremities of the face and the flipper here. And we saw some ulcerative lesions. So this is the bottom right, that white arrow is an ulcerative lesion on the fluke. And then another big thing, but wasn't necessarily a big thing for this animal, but that I want to talk about in this One Health picture is this picture on the bottom left, which we use a grease pencil. It's used in cattle operations a lot to, these orange marks on the manatee are where we have outlined a scar from a boat strike. Yeah, multiple boat strikes. So in this instance, if you can see the different colored circles, each different color is actually indicating a different boat strike event. And we know that because of these parallel lines. A great example is this, the yellow circle. So you can see 4 parallel lines and 1 perpendicular line right in going across those. And that is from a propeller, the perpendicular lines, and then the skag of the boat is the, the parallel lines, and then the skag is the perpendicular. So if you have multiple different patterns like that, you can assume that those are all different boat strike events. So I want to take just a second to talk about boat strikes in manatees. So Florida, the Fish and Wildlife Conservation Commission actually put out this paper a few years ago about watercraft related scars in manatees. And I was so blown away by these numbers that I try to share this with like every class I teach, because they found that 96% of adults had at least one scar from a watercraft related injury, 70% of subadults, and 34% of calves, like 1-year-olds, had scars from boats. And then 1 in 4 adults had been hit more than 10 times, and 5 adult carcasses that they actually had in hand had more than 40 strikes. So this really highlights the sublethal effects of boat strikes in manatees. So that's not always an animal gets hit by a boat and immediately dies, right? That can happen. But the cumulative effect of being hit multiple times can really lead to the detriment of the animal. And this is a population level problem in these species. Okay. So sorry, these are the graphic pictures I mentioned. It doesn't get any worse than this. So internally, so we always move in an exam from external to internal. So internally, this animal had really interesting findings that led us to, we actually published this case because it was so interesting as a sort of multifactorial cause of death for this animal. So on the left, you can see a piece of the aorta, and hopefully you can appreciate, it is cut a little oddly, but you can appreciate this narrowing of the aorta right in the middle of that piece of tissue. And that is what is called aortic stenosis. So that aorta is abnormally narrowed, which would have affected blood flow in this animal. And actually, in this case, led to what you see in the middle picture, which is concentric hypertrophy of the heart. So this is the left ventricle, which the aorta comes out of, right? So the left heart pumps blood out of the aorta. And if the blood can't get out of the aorta properly, it leads to a backup in the heart, and the heart has to work harder and harder to push the blood out. So the heart is a muscle, and the harder that you work any muscle, the bigger it gets, right? That's why we work out. So the heart in this case, though, was abnormally thickened, those walls. So that is abnormal. And it also led to some pulmonary edema. So the blood, the whole system backed up even farther to affect the lungs as well. Okay. But the reason that I really want to share this case in this example is that we also, as soon as we opened the esophagus of this animal, we found a plastic bag. So you can see in the top image, that's a part of the esophagus with the plastic bag still in it, and the plastic bag removed was kind of twisted up. And then untwisted in the bottom right, you can see it's basically a common grocery store bag. And we were able to show through histopathology, so that's the microscopic examination of tissues, that the bag hadn't caused much lesions in the esophagus, but based on our examination of the stomach contents, there were none. There was a lot of fluid. The animal had ingested a lot of water and fluid and a little bit of debris, but it had no normal gastric contents throughout the whole gastrointestinal tract. It really had no normal contents. So manatee GI passage time is like 7 days or so. So we think that this animal probably had not eaten for at least 7 days, and that plastic bag may have been preventing that animal from being able to eat. It's possible that it also just was so sick that it wasn't eating for a while, and ingested this bag in the last minutes before death, but it seemed like it had been there for quite a while. So this is a great example of, we're going to talk a little bit at the end about what we can do and reducing our trash and our waste in a One Health light, because marine debris affects everybody, the entire ecosystem. So just every case that we're going to talk about has these sort of highlights. So this animal experienced both natural and anthropogenic stressors, and underlying conditions such as cold stress in this case probably increased its susceptibility to the threats that it experienced. So marine debris is an issue across many different species, not just manatees. So in this case, on the left, you can see a bottlenose dolphin in our area. And we talked a little bit about entanglement earlier, and we'll talk about it a little bit more in this lecture or talk. But you can see this dolphin has those blue arrows are pointing out erosive lesions around the rostrum or the nose or mouth of the animal. And that we know is from an impression from a net being wrapped around the mouth of that animal. And that dolphin had other signs of having drowned in a fisheries net, which is not uncommon in this area, unfortunately. And then the bottom right picture is from a sperm whale. So this is the opened stomach of a sperm whale. And inside you can see just a huge wad of longline fisheries. So the spread out bit is at the top right. But this little scale bar in the photo at the bottom is 15 centimeters. So you can see that the length of this debris that this animal ingested is probably like 3 feet worth of debris. So that's a lot in this animal's case, and we're working on a sort of large case report right now about sperm whales in the Gulf of Mexico, of which a couple of the cases had large amounts of marine debris that they had ingested, which were theorizing could be related to decreased prey availability in the Gulf of Mexico. Okay, so another case that I want to walk through covers a couple of different areas, which is really great. And this is the one that we mentioned right before this with Dr. Moore, who was talking about freshwater exposure. And this animal's name was Allie. So Allie, we got a call, not me. I was not working at Dauphin Island Sea Lab at this time, but the Dauphin Island Sea Lab got a call that there was a dolphin stranded up where this red dot is in this picture. So this is Perdido Bay. So Gulf of Mexico is here, and then Perdido Bay comes all the way up here, and it goes into some rivers and small lakes. And the dolphin was in a small lake that was connected to this whole system. Do you guys remember what that term is when they're in an area and they can't leave out of habitat? So that is one type of stranding where an animal is out of its normal habitat and isn't, for some reason, it's not going back to where it should be. And the salinity in this area was very low compared to what it should be in their normal habitat. I think it was 10 or so. I can't remember off the top of my head.
I think it was, yeah, we had a huge, see previous, and in the introduction, I was talking about this huge amount of fresh water that we get that comes through. So we get huge fluctuations in salinity, and I think it was about 15 when the animal was there, but it varied even during the time that the animal was there. But in any case, it's low.
Okay. Yeah, so normal for anyone who's not familiar, in like the Gulf where it's fairly consistent, the salinity is more in the 30s parts per thousand. But in this case, it was more like 15, so like half of what would be considered normal. Although estuarine dolphin populations are used to more fluctuations in the salinity. So anyway, in February of 2016, there was a phone call that came in about this dolphin. And the initial response was, okay, let's see how the animal does. Can it get out of this area on its own? And there were actually 2 dolphins at that time, but I'm going to focus on the one that ends up in this case report. So February, let's just watch, monitor the animal. If it's eating, doing well, we don't really need to intervene. So next slide. So what are we concerned about in this case? And you guys have gotten multiple introductions to freshwater already, and Dr. Moore's talk did a great job explaining the pathogenicity of freshwater exposure. But for everybody online, basically dolphins, their skin, their whole body, their physiology is meant to be in higher saline waters, anywhere from really like 15 up. So when they're exposed to lower salinity for long periods of time, their skin can thicken, it can ultimately ulcerate, so they lose layers of their skin that they can slough. They kind of take on this pale appearance at first, and then they can develop these larger lesions like you see on the bottom right picture here, these big circular, they're called erosions, where the superficial layers of skin have started to erode away. And those can deepen and involve the blubber. They can start getting growths of like fungus and algae. And then more physiological things start to happen where they can get cerebral edema, corneal ulcerations and corneal edema, and develop kidney disease, electrolyte imbalances like Dr. Moore mentioned, and ultimately they will die from this if they don't either move to a more saline environment. Okay. So back to this animal, they received another call about a few weeks later in March. And that was a call that the homeowner said, hey, these dolphins are still here. So the team went out and assessed the situation again, and the animal was no longer foraging in the area. So the decision, and there were some potential freshwater lesions now at this point. So there was a bigger concern for the animal's welfare at this point. So just like the manatee case I mentioned earlier, now it's, okay, what can we do? Do we start a rescue intervention? So the answer was yes. So the animal, they ended up going in March of 2016, capturing the animal. So we did a whole talk earlier about capturing, and yes, this right here, woman of the day, is Dr. Moore. So she assessed the animal, and they ended up putting a satellite tag on the animal to be able to track its movements. So we talked about that a little bit later, and it's shown that if an animal survives like 45 days or so after release, they typically deem that a successful rehabilitation and release back to the wild. So that's one of the reasons, is to see what do they do after release, and do they live for how long after release. So At the time of rescue, the animal was thin. So we talked a little bit earlier about body condition in dolphins. So this animal's a little skinny. The top-down view is a little harder to assess, but the animal is thin. The skin is pale and sloughing, and then she did have superficial skin lesions and then corneal opacities, so corneal edema in this case. They did biochemistries and looked at the electrolytes, and the animal was, what are the electrolytes that might be off if an animal spends time in fresh water? Sodium chloride, yep. So she was hyponatremic and hypochloremic. And do you have anything to add since you're here? She had no idea that we were going to do this.
No idea you were doing this. Oh, go ahead.
Okay. So the abnormalities were small enough that they deemed this animal could be released and would likely survive if it went back into a normal saline environment. All right. So they took the animal from where it stranded up here all the way down to here to release. And so the salinity in that area is much more normal. And if you put an animal back into a normal saline environment, in theory, the skin should go back to normal and it should improve, right? Okay, so May 8th comes along. The animal, this is a photograph from that day. She's doing good. You know it's her because she has that satellite tag, but also the dorsal fin is unique in those animals. So doing well for a month and a half after release. But then unfortunately, she was found dead and called in on the 18th of June, which was, and right around in down here in this area. So on necropsy, same as the manatee, we're gonna do an external exam first. The skin lesions were found to actually have been much improved. So the picture on the left is from the time of the capture, and the picture on the right is 85 days later when the animal died. The skin lesions look much improved. They're still there. She's still like maybe a little bit pale and has a little bit of sloughing skin, but she looks pretty good. The eye lesions, so the corneal opacity had actually gotten a little bit worse, which is interesting. It's possible that even, so like the normal treatment for an animal's corneas like this is actually hypertonic saline. So moving to a hypertonic or a higher salinity area should have improved the corneal edema, but it got worse. So it's a little unclear. Maybe it just wasn't better enough for it to really improve in the amount of time that it was. The salinity still wasn't like high. It was just like a little more normal in the area where she was released. Or it could have been something separate, like trauma. It's really unclear why the corneal edema hadn't improved. Okay. And then there were findings that suggested that this animal actually probably died from drowning in a fisheries net. So there was froth in the airways, impressions of nets, just like I showed you from that other dolphin on her rostrum, melons, and fins. This one is actually, I believe, from her toe. So she was towed with a line at one point. So you have to be careful when you're assessing human interaction lesions and fisheries interactions lesions. Know what might have been iatrogenic, like you caused it. But she did have other lesions that were suggestive of a net. And then the real kicker here was that in her satellite tag, they found a piece of net, which was later identified as a piece of a shrimp trawl net. So dolphins in this area love shrimp, and they love to follow boats and swim into nets and eat out of them, and sometimes it's fine and they get a nice meal, and sometimes it just doesn't end well for them. So this animal was determined. I think there's a little. Yeah, so this animal was determined to have died from the like scientific term for it is peracute underwater entrapment, which is basically drowning in these animals. So again, this animal faced both natural and anthropogenic stressors, a One Health case in the sense of the freshwater and the fisheries interactions here. And it was just interesting to note, though, that we talked about, can animals survive freshwater and improve? And this is a great case of yes, they can. If she hadn't been taken by the shrimp net, she otherwise was doing really great.
So in every case, when we do a necropsy on one of these animals, we take teeth. And you guys know why we take the teeth? Yes, we age. So if you're familiar with the way you age trees, you know, cut them and you know, it doesn't work if you cut a dolphin that way, so you have to take the teeth. Yeah. But only from dead dolphins. But one of my graduate students who actually just defended his dissertation just a couple weeks ago, so is now Dr. Matt Hodenbosi, was in charge of this work. Yes, thank you, and on his behalf. But he took one of the teeth, he sliced it, did the aging analysis on it. But what we also did was use laser ablation ICP-MS, which is inductively coupled plasma mass spectrometry, which is something that we do in our lab to do metals analysis and some other kinds of analyses. But you also get to play with lasers. That's the operative word there, which is really fun. But what he did was he ran a transect perpendicular to the lines of growth in the teeth that allowed him to actually measure the concentrations of a whole suite of different metals at different elements in the tooth. And so our idea was, ooh, this might be an interesting case that would actually allow us to trace those links between the freshwater inputs and some of these metal contaminants. And so we used, and this is all in that same paper that Jenny was just telling you all the results from. And so it allowed us to do this sort of as a first time case study to see if this really works. And so what I'm showing you in the top panel here is just two elements, two metals that we pulled out. One is barium, which is shown in black, and the other is lead, which is shown in gray. And barium's important because it turns out that barium is an inverse salinity indicator, which means that you tend to have higher barium concentrations in riverine water, and it falls out as you move toward more marine systems, right? So what you should expect to see is an increase in barium where you have an increase in freshwater concentration. And so what Matt did was then plot the barium, and then also lead here is just an example of one potentially toxic or harmful contaminant that may be associated with freshwater. And then plotted that against this, in this case, it's distance along the tooth here, but that distance along the tooth allowed us to line the data to the dolphin's age. And so we actually have, it's kind of hidden here, but up here at the top, because the zoom thing is kind of hiding it, but up here at the top, each of these dashed lines is this is before the animal was born, and then this is a year of the animal's life. So this animal is just over 3 years old. And what you'll notice is that there are these distinct annual peaks. And it turns out that freshwater conveyance to our system, all this freshwater that's coming down, tends to, not always, but tends to be seasonal. So we tend to have higher discharge and higher freshwater input during the winter, spring, and lower during the summer, fall. And so what we think we're capturing here with the barium are these annual peaks in freshwater discharge. So this tells us a couple things. First of all, that not only did this animal have this out of habitat circumstance where it was, and we can see that actually here right before it died, it had enough time to put some barium in its teeth that reflect that increase. That they're not only experiencing this in an out of habitat situation, but our animals in our region are experiencing these intense increases in salinity on a regular, at least seasonal basis. And in this case, we also saw those spikes associated with a spike in lead. Now, if you compare this to other metals, which actually this became the basis of Dr. Hodenbody's dissertation, we can actually see that conveyance of that fresh water bringing certain metals, right? So then again, we're talking about cumulative effects as well. So not only all these different stressors that we already just talked about that are affecting this animal, but potentially at least seasonally, and whenever it's experiencing these high freshwater discharge events, it's also likely getting more contaminant load as well. And you can actually align this to those discharge data. So that's what the second panel here is showing you, which is the flow. And you can see that barium peaks are aligned to the peaks in discharge, and those are also inversely related to that salinity. So when the freshwater goes up, the salinity goes down, and the barium goes up, and the lead goes up. Does that make sense to everybody? cool or not cool, depending on how you look at it. But yeah, all this is published, and our vet at the time was Dr. Alyssa Deming, if you're familiar with her. So
It's at Pacific Marine Mammal Center. for
Yeah.
anyone.
And so we want to take a little sideline here and talk just a little bit about freshwater exposure. I know Dr. Deb talked to you guys earlier a little bit about, you talked about the Bonacary spillway, I guess, and did you talk about the 2019 UME? Okay, so I'll keep it limited, but for folks online who might not have been here for that, as I've already said, our system has really high freshwater inputs, and it's typically seasonal. Again, not always, but typically. But we're also seeing that this is likely increasing due to a combination of climate-related factors that are increasing snowmelt and precipitation, but also that's interacting with sea level rise. And then we have a number of these activities that are going on in our watershed that are potentially diverting freshwater, or in this case, a restoration project or a number of restoration projects that might increase the amount of freshwater delivery as well. So up on top, did you show this panel previously?
Go ahead.
Okay.
Let's review it.
All right, so this is the just a review of the openings of the Bonacary spillway back all the way to the 1937. And what you're looking at is the number of times that the spillway was opened over years up to 2019, because this was published around that time where we had this record opening. But what we're seeing is an increase in frequency with which the spillway is being opened, as well as how long that spillway is open. And if you're not familiar with the Bonacary spillway, this is just one of the spillways in Louisiana that was designed to keep coastal areas from flooding. So it holds back the fresh water. One of the problems with that, though, is it also holds back the natural sediment delivery that happens. So in the Birdfoot Delta, so in the areas downstream of the Bonacary spillway, which is there to protect human development and prevent flooding, those areas are being, unfortunately, sediment starved. And so we're not having our natural processes of sediment delivery when that fresh water is coming down, but we're still having erosion, we're having sea level rise, we're having subsidence. And so we're net losing land in this area. And so the idea behind the restoration project, and several of these that are planned, the biggest one being this Mid-Barrier Bay sediment diversion that has made a lot of news, a lot of attention, is the idea to pull out one of these obstructions and to allow the spillway to open and redeliver that sediment load. So the idea is this will be a restoration project, and a lot of these projects are done in the name of either protecting urbanization, protecting human development, or in this case, doing the same thing, but from the other side, instead of protecting it from flooding, it's going to bring back the land, essentially is the idea. So the deal, though, is that we're going to have a huge delivery of river water, and we're also going to have lots of sediment coming down. And so animals like dolphins that have become more accustomed to a more marine environment are now suddenly going to be experiencing greater freshwater volumes than before. And it's going to be fairly sudden, at least periodically. So the bottom line with this project and others is that it's a high cost project. None of these things are taken quite lightly, but we ultimately don't know. We don't know what the effect is going to be on animals like dolphins. If you're interested in this topic, there have been papers published sort of modeling what the expected outcomes would be based on some of the information that Dr. Bloodgood just told you. But this is really contentious, and there's lawsuits and at this time, as far as I know, unless you have new information, I think all construction is halted as a result of the lawsuits. And there's arguments in favor of and also against on behalf of wildlife, on behalf of people on either side of the argument. So it's a really challenging question for our area about how to manage this issue and what's actually going to happen in terms of people and wildlife. I want to talk about this a little bit because this is something that our lab's been very interested in, and we don't ultimately know. I'll give a little segue and then I'll stop. We don't ultimately know what those thresholds are for effect for dolphins. So even that paper that we just talked about that told you, we would expect, take a dolphin out of a freshwater environment, you put it back into a marine environment, you should expect to see it healing. But we actually have very little documented data on, okay, at what salinity does that happen? At what salinity do we see the negative effects? And at what salinities and how long does it take for us to see the recovery? And so that paper was one of the few that actually sort of had a chance to document that recovery. And so one of the things that we're working on right now in our lab, we've got a paper that is so close, we're so close, but we've been trying to run models for indices to determine the duration and the magnitude of salinity changes and how long at certain low salinities dolphins would be at before we would see them strand, right? Before we'd ultimately see mortality. So this is just a little bit of that, of those data.
So I'll walk you through this chart here. So you have the number of stranded dolphins on the left side y-axis, and on the right side y-axis, we're looking at salinity and discharge. And similar to Dr. Hodenbose's work that was showed earlier, you can follow a couple of lines that I'm going to walk you through. The green, or well, let's start with the dashed blue. So the dashed blue line is discharge. So you can see water coming into the bay through that dashed blue line, followed by the green solid line is the salinity. So when you have an increase in discharge, here's a great example here in 2019, where you have high discharge and low salinity, high discharge followed by low salinity. And then what we did, and this is Christina Diaz-Clark, who was the assistant stranding coordinator at Dauphin Island Sea Lab, and now she's in Florida. So she created this chart and overlaid the strandings with the salinities. And the strandings are either in white if they did not have freshwater lesions, or in black here if they did have freshwater lesions on necropsy examination. So you can see that there are peaks in these black labeled freshwater lesion animals after a decreased salinity event. And the big one, interestingly, in Alabama was in 2020. So Mississippi had their huge unusual mortality event starting 2019. Alabama really experienced this interesting die-off of dolphins with freshwater lesions in 2020 related to discharge into Mobile Bay. And you can see just a few more examples on the bottom here of what freshwater lesions look like in dolphins. It looks really not fun.
So next we're going to shift to a case with, it's actually, different kind of case, but we're going to talk a little bit about forever chemicals. And forever chemicals are these persistent organic pollutants. It could be things like the classic ones that we used to always teach about were DDT and PCBs. And nowadays it's all about PFAS and PFAS. So we're going to talk a little bit about that. But the reason why these contaminants are of such concern is simply because they are persistent, because they do hang around in the environment. They resist degradation. And unfortunately, they're incredibly common. One of the big ironies that I think about every time I go out to eat is how hard, when I was younger, we pushed to get rid of styrofoam because it was so bad. And we replaced it with cardboard. And now we cover the cardboard in this nice stuff that allows it to hold water and grease. And it's all made out of PFAS, right? So we just went from one harmful persistent organic pollutant to another, right? And PFAS, this is the one we're going to talk about a little bit more today, but it's in all kinds of things that you use around the house. And I'm not going to tell you should feel guilty particularly, but you should be aware, right? And we can then start working toward hopefully reducing this. But most of your clothing, especially if it's any kind of flame retardant or water resistant, most fabrics around your home. If you like your microwave popcorn, it's tragic. Again, it's that stuff that they put to make the grease not go all over your microwave. That's PFAS. Common paints, and even my husband's a firefighter, the fire foam. Every time I go out and I watch him use the fire foam, it's so cool. I'm like, you're putting PFAS out, right? But that's how common this stuff is, and it's everywhere, right? And so we need to be thinking about, wrong computer.
Yeah.
I did it again. Be thinking about, what's happening then in terms of, again, this is something that not only is at the base of our food web, but is moving up food webs, accumulating in marine mammals, and also could be accumulating in people. And so this is some work that we did with collaborators, David Halla and his recent student, Raina Nolan, who is also now Dr. Nolan. And we did work looking at everything from oysters to a variety of fish species out in the Gulf, and specifically in Galveston Bay. And then these are dolphins primarily from right here off the coast of Alabama. And we looked at liver, which are the bars shown in red, and mussel in all of the fishery species, but this is blubber for dolphins in the blue. And we found, as you might expect, as you move up the food web, you do see these high loads. And you notice there are a couple of split axes here that's showing you these jumps in the concentrations with the highest values at the top predator, which in this case are the dolphins. And so again, they're swimming where we're swimming. And this is in the water, right? They're not eating popcorn. They're not, using paints. right? But this is what's in their water. It's getting into their food resources and getting into the dolphins. They're swimming where we're swimming, they're eating what we're eating. Very likely, this is also happening to us in our coastal waters.
Other.
Okay, so now we're gonna talk about another interesting consequence of freshwater conveyance, and that is pathogen exposure. Yeah, I mean, I can go through this. We have till 4:30, right?
I just want to have a few minutes
Or
before
five.
we
We
get
have
John.
till 5, yeah.
Just a few minutes before, so.
He starts.
We're gonna be long-winded.
Yeah.
No,
We'll
we'll finish.
No, I'm
Yeah.
not rushing you.
I think we'll be done in 10 minutes.
Yeah, I'm not rushing. I just.
Okay. Okay, so next slide. So pathogens as a potential consequence of fresh water. You're good. Did I do it right? Yeah, that was great. Okay, so we're going to talk about 2 cases of erysipelathrix, which I talked a little bit about earlier today, but I didn't want to give you too many details because of this example that I want to talk about here. So we had 2 dolphins in 2020 that stranded very close together. So one is in this yellow dot and one is in this green dot in both space and time. So one was in February and one was in March of 2020. And this was during that freshwater event in Alabama that we showed on the chart just a couple of slides ago. Okay. So both animals died of acute erysipelathrix sepsis. So erysipelathrix is a bacteria that I'll talk a little bit more about in a second, but we know that it was acute because this one animal, they were able to take and do CT and MRI scanning of the dolphin, which is really cool. And that we have partners at Auburn. So this is a deceased animal, but you can still get a lot of great information from these advanced imaging of carcasses in addition to live animals. And on the right here, you can see some of the necropsy findings, including gas accumulation in the brain that leads to these sort of gaps formation in that brain tissue. And that's related to gas output from this bacteria. And based on that and the necropsy results, as well as histopathology, we were able to say, okay, this is a bacteria that's causing this sepsis and death. And then follow up bacterial PCR, which is polymerase chain reaction. It's a diagnostic tool that we can use to amplify organisms and figure out what they are. And sequencing, we were able to prove that the bacteria, the culprit, was erysipelathrix rhusiopathiae. So it's this kind of beautiful bacteria. This is a stain that turns the bacteria this sort of reddish color. So erysipelathrix is a zoonotic pathogen. So if you're not familiar with that term, it is like the epitome of One Health because it means that it affects all organisms and it can affect people. So in this case, it affects dolphins, but we know it's a primary pathogen in pigs and actually some like turkeys and other animals, but it can also affect people. So the pictures on the right here are sort of the classic presentations in those animals. So it's also called diamond skin disease because it causes these characteristic diamond-shaped lesions. They don't always look like this, but this is like the textbook example. And you can see the diamond lesion on this skin on this pig here. And then in people, it can just cause really terrible, painful skin lesions typically, but it can advance to cardiac disease and even death. So it is a great example of a One Health pathogen. So we had an REU student, which is the research experience for undergraduates, come and work with us. And now she's at Mississippi State. I don't know. Have you met her? Okay.
She's great.
You need to meet her if you haven't. So Kaylin was a wonderful student and came to us for a summer to do research. And she looked at these 2 cases to help us figure out where this bacteria came from. So she did sequencing analysis and this image, it's a little blurry for some reason, but the image on the right here, you can see all these little icons are animals. This is a phylogenetic tree where things that are next to each other are more highly related. So the two dolphin isolates are here in blue. They were like 99% the same. The two Erysipelathrix isolates from the two different dolphins were basically the same. And they were most closely related to, interesting, like a sea otter and mangrove sediment, but pigs. Our big takeaway here is that there were a lot of closely related pig isolates in this case. So she published the paper about this, and you can go to the next slide, that's fine. So this is, I think, a great example of freshwater bringing not only contaminants, but also pathogens into our waterways. And we were able to trace this bacteria back to feral pigs in Mobile Bay. So they're an invasive species. They hang out around the riparian zones, which is like right next to the waterways. And this pathogen is transmitted through feces. So they're living, and they're all happy, but they're pooping in the water. And we're doing some work now looking at markers of bacteria in the water that are showing that we can look at other species of bacteria to show that Erysipelathrix rises and falls with freshwater conveyance into the bay. Okay.
And I didn't realize we weren't setting out to demonize freshwater.
Yeah, I know.
Just become a takeaway. But just want to kind of wrap it up here by hopefully not depressing you too much, but getting you to think more holistically. This is actually a poster that was put together by one of my current PhD students who's actually going to be defending in about 3 weeks. So you can look out for her advertisement, Anya Brown. But this is a summary of her project, actually, which is a really great example of a One Health project. But I wanted to show it to you because if you think about all the cases that we just showed you, I mean, really what we're doing is we're putting together all these metrics of water quality, essentially, with metrics of health in the animal, and helping to tell this whole story about how we have links between what people do and the resulting effects on our water quality, on the habitats, and on the food resources for the animals that live there. And in this case, how those dolphins are being affected and responding. And then we can think more broadly about what that means for our own human health. So I think this just does a really nice job, sort of illustrating what we were talking about at the very beginning, and how this is so multidisciplinary and transdisciplinary. And I started out my career, I did not start out with marine mammals. Marine mammals sort of happened to me, but this is my bailiwick here. This is my passion, is the water quality side and understanding human health from that side. So it's been interesting, good example. And so I also like to wrap up with this slide, and I wholesale stole this off the internet, and I've not been able to find a citation. So if you're out there somewhere, my apologies. But I really like this because it also puts what we know into, just reminds us to put it into this framework of also our changing environment. And it's got the 4 major factors here that we think about when we think about changing climate, because all of this stuff that we just talked about, all of this is going on against this background of climate change anyway, right? Anytime we talk about any of these environmental stressors. So we've got rising temperatures, we've got, these more extreme weather conditions, which definitely we know that's going to affect our freshwater entry. We've got sea level rise, we've got CO2 levels changing, and that is necessarily impacting all of these factors in the environment that we know are linked to human health factors, right? And some of them, a lot of them, we just gave you examples of today that we're already seeing happening in our systems, right? So we need to really be thinking about this. We need to be thinking collaboratively, right? Like we're already, and this is where science is headed anyway. So One Health is just a great part of that, right? But we need that kind of research. We need you guys to be thinking holistically and collaboratively. We need funding that's thinking holistically and collaboratively. I love giving this to funders. So if any of you are secretly rich, talk to me later. But we know, and so ultimately because we know healthy oceans bring us healthier communities. We know this. This is not a surprise, right? Whether we're talking about wildlife or we're talking about people. And we know it's the basis of our healthy coastal economies, which trickles hopefully up. And we know if you live here on the coast, you all raised your hand at the beginning and told me how important this is, right? This is part of our cultural heritage here on the coast, being able to go out and see the wildlife and live in this environment. So we want to wrap it up by talking about some things, and on a positive note, right? Talk about some things that you guys can do to hopefully make a difference as well.
Yeah, so for World Ocean Day, we're gonna give everybody here some tasks to sort of go home and take away what we can do to help the planet and the ocean here. So outreach and education, we talked a lot about today, and Dr. Moore, I think, did a really inspirational talk earlier, getting us like, I wanted to go out and like apply to be a congresswoman today. So I think she did a super job with that. And that's one of our points here too, is spend some time getting to, you know, try to affect policy and get your opinions and your scientific brains out there. We need more scientifically oriented people making decisions in at the upper levels of our government. We also have, I think, things like this are really awesome. So the Sea Lab created these signs and gives them away for a donation out of, yes.
Point of clarification though, we actually didn't invent the sign. This
Oh,
was
yeah,
actually an
that's
FWC created sign, but when we started our sighting network, we piggybacked on their design because we thought it would be really important to have common branding across the coast. So no matter where you are, whether you're in Florida or you're in Texas, you can see the same sign and maybe it'll just have a different number.
Yeah,
I
so,
don't wanna take
no, that was a good point.
credit.
So these signs are given away for a donation for people to put around their waterways is the especially important place, so that people know that there are manatees in this area and other wildlife. Coastal cleanup, get involved with your local organizations. We showed a couple of cases that involved marine debris today, so it's really important to use less wasteful products, and also if you do need to use them, make sure you clean them up and get them where they need to go after you're done with them. And other policy things too. So working with NOAA and the federal government on changes where we can. And then, so I end here because this is my new passion, is working with stranding response organizations. So I'm based at a university now, but I work with the stranding groups based in New York. And we are trying to just build relationships regionally, nationally, and internationally, hopefully one day, and build capacity for these organizations to do these advanced diagnostic techniques like CT scanning, working with veterinary students who don't often get opportunities to work on marine animals. Like, have you guys had a dolphin come to your university? This is what I'm like, yes, let's try and get dolphins so students can learn hands-on. And I bring students out to large whale necropsies with me too. So just integrating all of these multidisciplinary, collaborative responses, I think we can do a great job of addressing ocean health. I always end on this slide. So this is the takeaway of just, if you find a stranded marine mammal, the number to call, some points to follow, and the takeaway point of never pushing a marine mammal back into the water. Always just call the appropriate response agency. So thank you. And
I think them.
yes.
Yeah, a lot of people who made this work possible. So it was really nice to get a chance to speak to you all.
Thank you both so much. This is just a fabulous opportunity, I think, to tie it all together on World Ocean Day and the One Mississippi River Days of Action, and at Marvette, Mississippi, and Mississippi Aquarium. We can't forget about that, and all the great instructors and people that are here with us today. We have some phenomenal people on our Zoom too that are related. We've got brilliant scientist. I'm going to call her out, Stephanie Seneff, who wrote Toxic Legacy. She's a foremost scientist on the glyphosate issues, and I'm sure her head is spinning right now. And she's probably going to want to collaborate with you guys on this glyphosate issue, but we'll go there another time. So let me check. We have a few. There might be some questions here. There's okay, yeah, everybody's thanking you for all. Oh, I was muted. Oh, sorry. Stephanie. Yeah, I think they heard me. Okay, so I think that was early on. Do, do, does anybody have any quick questions, especially you on Zoom? Because we have another presentation. Any questions?
We can take, yeah, go ahead. Oh, boy.
Okay.
So I read a paper last year about, from A&M about there being like pharmaceuticals, like fentanyl, dolphins and Gulf of
Yeah.
Mexico. So I was curious if the freshwater discharges increase those pharmaceutical tendencies and concentrations and like what impact that has on them.
So get back with me in about 3.5 years. I have a PhD student who just started. She actually will matriculate in the fall, but she's already working with me, and we're working with folks at A&M. And we are doing some case studies with dolphins. We're also doing work with manatees, and we're adding in some of the pharmaceuticals. So the short answer is we don't know just yet. But if you look at, I don't know if you were reading, there was a study out of Florida, I think Bossart's group, that found that 80% of the dolphins in the study that they were analyzing had, in that case, it was antibiotics.
Yeah.
So they had antibiotic resistant drugs as well. And so scary. Yeah. I mean, it's intense to think about how just widespread, these contaminants, these drugs are. And we've done a little bit of work. I don't know if you noticed on the water quality slide, but we had artificial sweeteners in there. That's another great way to test for human influence because
Aspartame.
yes, aspartame, those don't exist naturally, and they're so clearly associated with human wastewater, human footprint. So yeah, and if Dara Orbach's group also, if you're interested in that, out of Texas, has done some work over there as well. She's got a student who's okay, yeah. But for our area, we don't yet know.
Thank you so much. And then the second question was, do y'all ever utilize the dolphin whale 911 app? Is that like a good way to report stranded marine mammals, or is it preferred to just call?
I don't know that we've ever received a stranding from, I think that it's a good app and definitely use the app, but I don't know that we've ever received a stranding report from the app. So for cetaceans, the easiest thing to do is that 1-866 whale help number, because you can go anywhere. It doesn't matter where you are in the country. You call that number and then you just push a button for your state that you're in. That's super easy. Have you received reports through the app?
I don't know, that protocol came from New York that was called in and then went down to you all.
Oh, yeah,
I don't know if that was the 911.
maybe.
Maybe for live, like actively live strandings. I'm not sure. That's an interesting question. Yeah.
Just with the cold stress in manatees, how we're seeing that impacted with them not being able to migrate back down to the warm climate with climate change, and how that's
Okay, so can you repeat it? I was still thinking about whale help.
I was wondering how the cold stress in manatees not being able to migrate down to the warm habitats is affected by climate change.
So it's not that they're not able to migrate, they are able to migrate.
Okay.
And the vast majority of animals that come through here, they go back and they seem to make it just fine. I think there's some important things to know, and that is that we're having more animals here now, and the animals that we do have here now are staying longer in the season. We have a couple of papers on this, and we do see growing evidence that they're probably finding ways, at least in some years, particularly mild years, to overwinter. We don't fully understand the mechanisms by which animals are not, like, why are some animals not making it? Those rare ones that don't. And this past winter, I don't know if you guys know this, but it was like crazy here. We had cold. This is not why I moved down here. I'm not originally from here. I came here to be warm. It was cold. It was sustained cold, and we had snow for like a week. I was completely indignant. Right? We had sort of a manatee apocalypse. We had a record number of live manatee strandings. We had a record number of rescues, and we actually had a record number of successful rehab. And we successfully captured and took a couple of cold stress animals to SeaWorld Orlando, where they have a critical care facility, and they've already also both been released. We actually had the first successful rescue of the year right here in Alabama waters for manatees. So we don't understand exactly why some of them, what's happening when they're not migrating. Like, is it that they're not getting a cue? I strongly suspect that we have many, many more animals that are now going even further west because we have manatee sightings all the way down, literally right to the border of Texas and Mexico that come in. So the further west you go, when the water gets cold, you have to kind of book it back. And it may simply be that the animals that we're seeing, maybe they were further west. It's just taking them that long to get to our area, and then they're just not making it back. It could be that some animals we've thought about, are there animals that have been maybe HAB affected and they're not getting the right cues and they're not making it back. And it could be that they're just doubling down on, hey, this is now home because these animals, and we know this from our tagged animal data, they're animals that are spending more time of the year here than they are in Peninsular Florida now. So they go back just for the coldest time of the year, and then they immediately come right back here. And so if you're spending most of your time somewhere else and you just have to migrate back to sustain that over winter, it kind of makes sense that some of these animals could then just be, weighing in the balance as this game theory, right? Maybe I can stay. And in some years they can. And then maybe some years when we have this crazy years like this past year, they can't. And we have animals that are caught, and certainly if we have a cold snap early in the season, we'll see that sometimes too. Yeah, that was probably more than you wanted to know, but.
Well, that's just fantastic. You guys, this is like way amazing. And then to have Dr. Deborah Moore here, if I would have known that, I would have had her on Zoom too. But anyway, it's been fantastic. So I guess we should take a little bit of a 10-minute break, and we'll try to get John Rusky set up, and then we'll go have dinner and maybe go catch up on some sleep. Thank you, everybody, for joining us.